Hi friends, in this we are going to discuss about the laboratory equipments that we are going to use in the lab and their uses. First, hot air oven. They were originated by Pasteur, that is Louis Pasteur, and oven uses dry heat to sterilize articles. We need to sterilize what are the tips, tubes that we are going to use in molecular biology lab. All those will be sterilized with this. Generally, they can operate from 50 to 300 degrees Celsius. And coming to incubator shaker. In molecular biology, incubator shaker is a device. As you can see here, there is an incubator. The device used to grow and maintain biological cultures in a cell. The incubator maintains optimal temperature, humidity, and other conditions as in the carbon dioxide, CO2, and oxygen contained in the atmosphere inside. That is, in this we are going to in the flask. You can see here flask. Within this, there will be optimum temperature, humidity, all those conditions will be maintained for the cell culture or microbial cultures to grow in those. Coming to centrifuge. Centrifuge is a piece of equipment generally driven by the electrical motor, that is centrifuge, and this that puts object in rotation around the axis, applying a force perpendicular to the axis, that is force perpendicular to axis. The centrifuge works using a sedimentation principle, that is, as long as it continuously rotates, there will be sedimentation will take place, that is, hard materials will be sedimented here, light materials will be floating. Where the centripetal acceleration causes dense substances to separate along the radial directions of the bottom of the tube. By the same principle, lighter objects will tend to move top. As I said, light will be moved top and the hard one will be settled down. Magnetic stirrer or magnetic mixer. Magnetic stirrer is a magnetic mixture is a laboratory device that employs a rotating magnetic field to cause stirrer bar. Previously, we used a stirrer to stir with our hand, like the stirring, continuous stirring. But with the help of magnetic stirrer, there will be some beads will be there. Magnetic bead will be there. Again, it's placed on here, and due to change in the magnetic field, there will be continuous rotation, so that there will be bead also will be rotation takes place inside the beaker. Rotating field may be created either by rotating magnet or a set of stationary electromagnets placed beneath the vessels in the liquid. PCR, it's one of the important equipment that one laboratory must have. PCR means polymerase chain reaction. The biochemical technology in molecular biology amplify single or a few copies of DNA across several magnitudes. That single copy is magnified 1,000, lakhs, billion times. There will be multiplication will take place. And it's developed by 1983 by Carey Mullis. Used in DNA cloning for sequencing, DNA-based phylogeny, and functional analysis of genes, identification of genetic fingerprints, used for forensic and paternity testing, and detection and diagnostic of infectious diseases will take place. As you can see, this is Pcr machine, one of the most important invention. And pH meter used to set the pH of the solution will take place. And thermocycler is also known as PCR machine or DNA amplifier, is a laboratory apparatus mostly used to amplify segments of DNA via polymerase chain reaction. And this is also a thermocycler, nothing but a PCR. Micro oven used to melt the medias. For example, for gel electrophoresis, we need a micro oven. Elisa Reader. Elisa Reader measures the quantities of the color differences in the 12 wells of the plate. Elisa Reader or microplate readers do spectrophotometry. They emit light at one wavelength and measure the amount of light absorbed and reflected by an object, such as protein. Spectrophotometer measures the ultraviolet and visible light. As you can see here. Electrophoresis unit. A technique of electrophoresis is based on fact that DNA negatively charged and as neutral pH due to its phosphate backbone. For this reason, when electrical potential is placed on DNA, it will move towards the positive pole. You can see bands in the gel like